Hello everyone, we will continue the structure of bacteria. My name is Dr. Sharad Deshmukh from SS Jaiswal College, Arjuni Morgaon, District Gondia. Today we will going to see about the structure of bacterial capsule. So you know that the bacterial cell, which consists of the cell envelope, and in some bacteria they may possess one more outer covering that we refer as a capsule. So what is the bacterial capsule? It is a viscous substance forming a covering layer or envelope around the cell wall with definite margin. This is known as the capsule. It is not present in every bacteria, but some bacteria may have this structure or some may not have this structure. So every bacteria is not capsulated, or every bacterial cell is not the capsulated. Some may or some may not have. But if it is with the definite margin, then it is referred as the capsule. So this capsule can be visualized by the light microscope by the special staining technique. You have to use the special stain to visualize them under the light microscope. Now this bacterial capsule can be classified mainly into the two types. One is the macrocapsule, which is at least 0.2 micron thickness or more, and that can be visualized or that can be seen under the light microscope, that is referred as the macrocapsule. The second type is the microcapsule that cannot be seen under the light microscope because it is very small size, less than 0.2 micron thickness, and it can be demonstrated only immunologically or you can say serologically, and that is referred as the microcapsule. So the two types can be classified, macro and the microcapsule. Here you can see the clear halo around the bacterial cell. You can see central bacteria surrounded by a capsule, and background is the dark one. Now in bacterial capsule, you should not confuse the another term, that is the slime. Sometimes this capsular material is so abundant that the many cells, that number of bacterial cells are embedded in a matrix, and the material is referred as the slime. So just I have already told you the capsule means it is with the definite margin surrounding the cell wall, and this slime means the large number of the bacterial cells are embedded in the same matrix, that is referred as the slime, that is abundantly formed by the bacterial cell. So now let us see what is the chemical nature of the capsule. The major chemical component in the capsule is the polysaccharide or the disaccharide in nature. So majority of the capsules are polysaccharide in nature, and in polysaccharide it may be either homopolysaccharide or heteropolysaccharide. So one of the best example I have mentioned here, the Acetobacter xylenum, which possesses the capsule which is made up of the cellulose, which is the homopolysaccharide, while the heteropolysaccharide capsule, it is mainly seen in the Klebsiella pneumoniae. The next one, sometimes the capsule is also made up of the polypeptide. The best example is the Streptococcus species, which contain the L-amino acid, that is the polypeptide in nature. While in some cases, in Bacillus anthracis, it is a polymer of the glutamic acid. So this is the chemical nature of the capsule, commonly seen either the polysaccharide most commonly, or sometimes the polypeptide, or it may be the glutamic acid. So this capsulated bacteria, there are a number of organisms which are included in this group. One of the example is the, I have mentioned earlier, is the pneumococcus, most commonly given. But in addition to the pneumococcus, there are a number of bacilli or the bacteria, they are under this group, that is the Bacillus subtilis, Bacillus anthracis, Streptococcus pneumoniae, that is also referred as the Diplococcus pneumoniae, then Klebsiella pneumoniae, Haemophilus influenzae, Clostridium perfringens, Neisseria meningitidis, Pseudomonas aeruginosa, and some other bacteria like Acinetobacter species. So these are some examples of the capsulated bacteria. Now let us see what are the functions of the capsule, what they contribute to the bacterial cell. So one of the most important is they provide the protection to the bacterial cell against the temporary drying. How they can prevent it? They by binding to the water molecules, and they help to provide them the protection temporarily. while if the cell is the non-capsulated one, that can be easily killed as compared to the cells which are the capsulated one, because this has the ability to resist the drying at least for temporary period. Secondly, it is the most important function, that is it blocks the attachment of the bacteriophages. The bacteriophages which attack the bacterial cell, that phages which are attacking the bacterial cell, that can be prevented by the presence of the capsule. Then it is having the anti-phagocytic activity. So those bacteria which are capsulated one, they cannot be easily phagocytized, and this can be protected against the process or the phenomena of the phagocytosis, which is one of the preventive measure in the human being, and that's why they are contributing to the virulence of the bacteria, as they are the anti-phagocytic in nature. And another important is they promote the attachment of the bacteria to the surfaces. For example, the Streptococcus mutans, which is responsible for the dental caries. And the Streptococcus mutans is the capsulated one. It can adhere to the smooth surface of the teeth because of the secretion of the capsular glucon. And this capsular glucon is water insoluble. And that's why it is able to cause the dental caries in the human beings. So these are some important functions or the contribution of the capsule to the bacterial cell. Now let us see how it can be demonstrated. There are various ways we can demonstrate the capsule. One of the special staining technique for the capsule is the Anthony staining, and the his staining is used. Secondly, the most commonly used is the routinely used in the most of the laboratory is the negative staining, that is by India ink. The capsule appears as a clear halo around the bacterium, as the ink cannot penetrate the capsule. So it can be easily demonstrated in our laboratory under the compound microscope. Then serological method, it is particularly useful for the bacteria which are having the microcapsule, that is known as the Quellen reaction or capsule swelling reaction. So in this case, the specific antisera is used against the capsular material, and if the suspension of the bacteria is mixed with this antisera, the capsule will swell in presence of that antisera. That is known as the Quellen reaction or capsule swelling reaction. Then the Manuel's capsule stain is one of the special stain is used where the capsule appears as a clear halo between the pink stain bacterium and the bluish gray stain background. So background is the bluish gray and the bacterial cell is the pink, and the capsule will appear the clear halo. The background stain is the acidic stain, that is the Congo red, which changes the color to the bluish gray due to the pH, and the pink stain is acid fuchsin. So which are used in the Manuel's capsule stain, which can be used for demonstration of the capsule. So thank you for observation. I will see the flagellum or the structure of flagellum in our next lecture.